Hi, my name is Lauren. I am a senior at Beloit Memorial High School and a welcome intern at Welty Environmental Center. My project I did for GLOBE was a comparative analysis of tributary and main channel inlet ecosystems. What sparked me to create this project was this year I took an AP Environmental Science, and in doing so, we learned a lot about our local Lower Rock River and the pollutants that occur within it, specifically leaf litter that causes a elevated phosphate level, which then causes algae growth and in turn lowers oxygen levels and fish abundance due to that. I was curious how that just overall pollution in the Lower Rock River affects it as an ecosystem. So my testing I did was comparing Wooten Park, a main channel inlet of the Lower Rock River, with Leeson Park, which is a tributary of the Rock River coming from the same watershed, but previous to the general pollution. So Wooten Park is the park that I tested right by the high school. This is all my high school. And then Leeson is over here, and it is in a floodplain of Beloit. So that was also something I will take into consideration within my testing. So the questions I was curious about are how they sustain aquatic life, and just what does it mean for a healthy ecosystem, and what would that look like? I was curious about flow rate, water depth, conductivity, the riparian zone, water temperature, and dissolved oxygen. However, as one of my limitations to this project, I found out my dissolved oxygen was expired as of 2017, so I did take that data out due to validity and not wanting to have that as a possible barrier. As well, as I'll talk about in a minute, I set out traps to test the fish. We did have a lot of interesting weather happen during this time due to the winter to spring transition for Wisconsin. So I did take that in consideration of not knowing all the fish activity habits and how these affected them. So you will not see my total fish data in here either due to that. So for my research methods, I would go out once a week, starting Monday and Tuesday. On Monday, I would go out to each site, take air temperature using the GLOBE protocols, leaving it out for 2 minutes, checking it, 3 minutes, checking it, and 4 minutes, and then taking the average of that data, as well doing water temperature, following that same thing, but doing it 10 centimeters below the water surface. And then conductivity, which I took a sample of surface water with a beaker and then used the conductivity meter to test that, which I did calibrate previous to the start of this experiment. As for the traps, each trap I placed was using a tent stake tied to a rope, and I used a minnow trap with a 2-inch opening at each end, and then I baited it with a plastic Easter egg with holes in it with dog food, and then had the proper labeling with our Welty and then my personal information in case they are lost or found. Here is some specific pictures of the sites I worked at. This is Wooten, which is the inlet of the main Rock River, and then Leeson's. As you can see through this picture, there are some differences that I will get to in my physical characteristics as well. Some of the data I did find was the correlation between air and water temperature, which is nothing truly groundbreaking. However, you can see a spike where the water temperature is able to stay above air temperature, which was a little interesting, but that comes down to water being able to retain heat longer than air once it is collected. However, down in the conductivity, you can see a trend where the air temperature and the water conductivity have a similar pattern. And this is due to water ions, and as the air temperature is hotter, the water temperature gets hotter, and that causes the ions to move quicker, keeping the conductivity higher. However, you can see that the tributary was still slightly higher in conductivity, which was interesting. This dip we can account for through a rain that did occur right here. However, you can see the tributary still stayed higher. This is likely due to it being a floodplain. And when it floods, the water goes up into the river, or not the river, sorry. It goes up into the land, and then when it pulls back, it takes nutrients from the soil that help counteract that conductivity, keeping it higher. As for the physical characteristics I was talking about earlier, We saw at the main channel inlet, it was a consistent 2 to 3 depth standing water. And then there was moderate vegetation everywhere, from grasses all the way to the bank, small trees, large trees, and types of shrubs. Whereas at Leeson, at the Tributary, the water changed from 1 to 4 feet throughout the season I was testing, and this was likely due to the rains and that it is in a floodplain. And because of this, it changed from a rippling water to more of a rushing water as it went higher in depth. As well, possibly also due to a floodplain, the vegetation is very sparse. There was some moderate tree coverage everywhere. However, there was no small vegetation that was towards the bank or anything like that. It was significant leaf litter and trees, and that was it. So what I kind of pulled from all of this is that when it comes to ecosystem health, it really depends on what you're looking for specifically. They both have characteristics that you would associate with a healthier and a less healthy ecosystem. So what I concluded was the tributary can sustain a larger variety of life, specifically aquatic life, however, not nearly as large of a population. As well, on the other hand, the main channel inlet could sustain a larger population of fish, however, a smaller biodiversity within that population. As well, when you're comparing the entire ecosystem as a whole, the underwater versus the above water habitats were slightly different, as we talked about the vegetation versus those populations are kind of opposite, which is an interesting hypothesis to test further in the future. Some other future research I would be interested with those conclusions I found were I did catch a single crayfish at both sites. And originally I'd wanted to do crayfish, however, they only come out at 40 degrees, so I was not at that place at the time. So I would be interested in learning more about the indicator species and how they affect our ecosystems. As well, in our native area, we have invasive rusty crayfish that could also affect our ecosystems.